everyone. Welcome to MGBS 2025. We are delighted to have you with us for the Forces Today lecture. It's my great pleasure to introduce Mr. Abdelaal Shehata, an estimate of Fulbright alumnus and a current PhD student at the Department of Entomology and Plant Pathology at Auburn University in Alabama, United States. He gives us a lecture under the title Sequence Characterization of Soybean with Necrosis Virus SVNV from Field Isolates in Alabama. He received his master's degree from Auburn as well, where he focused on sequencing negative sense RNA plant viruses that belongs to the genus orthospotvirus, including tomato-spotted willet virus and the soybean vein necrosis virus. Furthermore, he conducted many studies on localizing this virus's protein in plant and insect cells, which allow us to understand their infection cycle within their plant hosts. Currently, he is working towards his PhD, and his research goals are to understand this virus's replication and infection cycles within their plant hosts, ultimately assisting in finding better management methods to prevent the loss of millions of dollars caused by these viruses in Alabama each year. In addition, he is also working on comparing the movement routines of a group of deserved plant viruses to understand how each virus moves within its host, which will provide new management methods. It's wonderful to join us, Mr. Abdul. Thank you and good luck.
Yeah, sorry for the interruption, but I would like to add something important about Abdelaal. Abdelaal actually is a member of MGBS team, and we started organizing such events together in 2017. And despite his busy schedule in USA while pursuing his PhD studies, he remains a very active member. He not only participates each year in MGBS with his research, but also makes significant contribution in organizing these events. So frankly speaking, Abdelaal really is a promising young scientist with whom you would like to enjoy working and collaborating. Thank you, Abdelaal. I would like to personally thank you for your efforts in MGBS, and I am looking forward to watching you in the MGBS and listening to your lecture.
Thank you so much, Dr. Ahmed. I'm really appreciative to the opportunity you give me every year and also contributing with the organizing the conference or the symposium with you every year. Thank you so much for your introduction, and I will not delay you anymore. I will start directly. So soybean is a very important crop in the United States. It's used mainly for textiles and biofuels, and also it's used as a food source for both animal and human consumption. In the United States, the production of this crop reached about $56 billion in 2023, which is a huge number of money. In Alabama, it is the first crop being planted, and you can tell it's very important for the country and for also other countries. When you look in the map here, we see the soybean production is mainly focused on the north of the state. However, in the south and also in the center of the state, we also still have some counties planting soybean. So unfortunately, soybean vein necrosis virus is infecting soybean, and that's not a good thing because this virus can kill the plant cells, causing necrosis symptoms, as we can see here on the figures. And the history of this virus, as you can see here on this timeline, it was first discovered in Tennessee in 2008, and later in 2012, it was detected in Alabama by Dr. Sakura. The symptoms of the virus, as I just mentioned, they comprise of necrosis, which is basically a term used in plant pathology to refer to cell death. So whenever you have cell death, you have necrosis, and it appears between the veins or along the veins of the infected leaves. And that's why the virus name is called vein necrosis. As other orthospotviruses member, this virus is transmitted by thrips in a persistent propagative manner. So basically, when the eggs of thrips hatch from their eggs, they feed on infected plants during the larva stage, and once they pupate in the soil, the virus uses this period of the insect being not active and then multiply and replicate within their bodies. After that, when these pupa hatch or emerge as adults, they feed on healthy plants and then they transmit the virus to those healthy plants. I would like to mention also that this virus, to be transmitted by the adults, it has to be acquired by the larva stage. So here I wanted to talk about the group that is called Orthotis bovaris, and I know it's a very long name, but basically it contains plant infected viruses. But this group also belongs to another bigger family called Bunyaviridae. This family has both plant infecting and animal infecting viruses. So these viruses are very cool to work with because they can cross kingdom. They can both infect the animal system and the plant system. And here I listed 10 main groups or strains of the Orthotis bovaris, and I highlighted those 5 here because those are going to be mainly discussed today. First, of course, I highlighted the soybean necrosis virus because it's the main talk today, but also tomato spotted boll virus, ambitious necrotic virus, and iris yellow spot virus, and capsicum chlorosis virus are also going to be compared to our SVNV once we do the sequence and the localization as we will discuss today. So these group of viruses have a tri-segmented genome. This means that their genome is composed of 3 segments. We have the S segment or the small, the medium segment or the M, and then the L segment or the large. So these 3 segments have negative sense genome. So basically on the S segment, we can look at here and see that we have 2 different open reading frames in a different orientation. So we have NSS in the positive sense, but N is in the negative sense. This makes this segment ambisense. And also on the M segment, we have the same. We have NSM and also GN and GC in the opposite direction. Makes it also an ambisense strand. But on the L segment, we have just one open reading frame, which is the RDRP polymerase, and it's in the negative sense strand. So what are these genes and what are they doing for the virus? So basically, the first one, NSS, it stands for non-structured silencing protein. And as you can tell, it helps the virus to silence the plant system and the plant mechanism defense. This allows the virus for successful infection. The N protein or the nucleocapsid protein is very important for the virus because it wraps around the RNA of the virus, as we can see here on this virion, and it forms something called ribonucleocapsid complex. This helps the RNA of the virus to be protected and not to be degraded. On the M segment, we have the NSM or the non-structured movement protein, and as you can tell, it is responsible for moving the virus from cell to another cell during the infection. The NSM protein of SVNV and other Orthotis bovaris groups, they belong to the 30K superfamily, which contain a conserved motifs and domains that helps them to function as movement proteins. However, Orthotis bovaris, including SVNV, they have a specific domains responsible for the tubule formation, and it's unique to the Orthotis bovaris, which is cool. The GN and GC and stands for the glycoproteins. They are found on the M segment, and also they are very important for the virus acquisition by the insect. So we can see them here on the outer shape of the virus, work as attachment to attach the virus to the insect style so it can be acquired by the insect. Finally, the L segment has the RDRP, or basically the RNA-dependent RNA polymerase, and is responsible for the virus to be replicated during the infection. So the distribution of this virus is very interesting because while it has a narrow host range, by infecting so far 10 or 11 host plants, including the new detection of this virus in peanut in Alabama this year. However, it's widespread. It has a very narrow host range, but it's widespread. We can find it in different countries. However, it was found first in the United States. Now it's found in Canada and in Egypt and in Pakistan as well. And I would literally die to get some sequences from these other places to compare them to the sequences that we have in Alabama, and we can see the diversity of this virus. Here also I wanted to show you a phylogenetic tree to show you the relationship of this virus compared to other Orthotis bovaris. So the first phylogeny tree here on the left, it's basically was generated based on the N protein. The one on the right was generated based on the RDRP protein. The one on the left here shows us just the Orthotis bovaris groups. However, the one with the RDRP on the right shows us the Orthotis bovaris to other also families that are different from Orthotis bovaris. So when we look into both these trees, we can see something interesting here. We can see that SVNV is only clustering or grouping with one virus called B necrotic mosaic virus, and both of them cause necrosis. However, both of them are distinct and different from other groups of Orthotis bovaris. We have basically 5 main groups, and SVNV is clustering, interestingly, along with B necrotic mosaic virus. We can see the same pattern on this phylogeny as well. I know it's very tiny, but here you can see B necrotic mosaic virus with soybean vein necrosis virus. And hopefully today we can reveal this association and why SVNV is clustering with being necrotic away from other groups of orthotusboviruses. So now to the important question, why we are studying SVNV. Of course, I mentioned before, but let's put them here in bullet points. So first of all, SVNV is a seed-transmitted virus. It's basically the only orthotusbovirus that can be transmitted by the seeds. And there's only some studies that showed that SVNV can affect the seed in content for the oil. It can reduce the oil content in the infected seeds. Therefore, it can impact the seed negatively. It also can impact the plants. SVNV has a phylogenetically relationship different from other orthotusboviruses, and that makes us interesting to be studied. Also, SVNV is always found in mixed infection, actually most of the times. So this virus basically needs company. It needs to be, and somehow it needs to be with other orthotusboviruses to help it to be more successful for its infection. And this also makes it interesting to study. And as we mentioned before, soybean is a very important crop, and SVNV is infecting soybean, so it's important to study this virus. And however, we don't have a yield loss for this virus yet because it appears late in the season. However, maybe it will become a potential threat in the future, so it's better to study it now and know about it. Also, SVNV is found in many countries because it's widespread, so it's interesting to study it now. Also, something interesting to mention is that in Alabama, in 3 years period, the symptoms of this virus and the incidence have increased from 31 to 82%. However, this looks scary, I know, but it doesn't happen every year. So some years we will have other infections, so we would have a lower infection of SVNV. So it depends on the weather condition and other diseases as well. But this is also a reason why we study this virus. So what are we missing about SVNV? So this virus, it was recently discovered, as I mentioned in the introduction, it was discovered in 2008, so it's a new virus. So we were missing two things here. One is specific to Alabama and one is for general. So the one for Alabama is that we didn't know the sequence of SVNV. We didn't know how it looked like and how it compares to other states. Is it similar or is it different? We didn't know. So sequencing the virus from Alabama and compared to other states is very important and would allow us to know what are the patterns that we have in Alabama. The second one, which is the general one, is that since this virus was discovered, it has never been localized in the plant cells. And if you ask me what is localization and why it's important for you, I will tell you that localization basically is the expression of the proteins of the virus into the plant cell or the host cell. So basically we have a plant cell here, as you can see here on this right figure. And when you express the proteins inside the cell, you can start seeing them somewhere in the cell. They have to be maybe somewhere here, like in the nucleus or the cell periphery. They can be in Golgi, or they can be just in the cytoplasm. Why this is important? Because if you know that this virus localizes in these locations, now you can make some predictions and some discoveries about why the virus needs to be there. Does it need to be there for the infection? Does it interact with something from the cell? And this is all important to understand how this virus behaves in the cell, which is really crucial for management methods in the future based on the molecular level. So the hypothesis of this work and the objectives are basically, we have two main hypotheses here. The first one is that the sequence of SVNV has likely undergone changes since it was originally sequenced in the study that was conducted in 2009, because it's been almost 15 years and we need to know if the sequence is evolving or not. Second hypothesis here is that the localization patterns of SVNV proteins is likely to be similar. However, it might not be identical to other characterized orthotusboviruses. So therefore, we have three main objectives here to investigate our hypothesis. The first one is to collect symptomatic soybean plants and amplify the first three genes, which are N, NSS, and NSM, and then sequence them using Sanger sequencing. After that, compare the sequence of the Tennessee, which was the first sequence that was deposited to NCBI in 2009, and to detect mutations and also construct phylogenetic trees that would help us to understand the relationship of our sequences. Finally, to conduct localization study on SVNV proteins in the plant and insect cells to understand SVNV behavior with the host. So basically, here's the workflow for the methods that we use. So thanks to Dr. Sakura, we collected 33 samples in 2023. And as when you see here on the map that we use for collecting the samples, we mainly collected samples from the north. And that also goes back to when I told you that Alabama produces more soybean in the north. However, we collected samples from the south as well, but we never went down to the south of Alabama because the symptoms of the virus are less prevalent in the south. And that was 2023. But in 2024, we also collected samples, not going to be covered today, but in 2024, we collected samples and we covered the 3 parts of the states. We were actually able to find few symptoms in the south of Alabama, but we haven't done sequence on them yet. So we have 2 groups here coming from north and from central Alabama. So after that, the samples were taken to the lab, the RNA was extracted, and the CNA was made because the virus is RNA and we cannot run PCR using RNA, so we have to convert it to a complementary DNA or cDNA. After that, the gene were amplified using the specific primers for N, NSS, and NSM, and then they were cloned in the PGID vector and sent for Sanger sequencing. After I received the sequence, all the sequences were analyzed using BLAST and also compared to the Tennessee strain to detect the mutations, and the phylogenetic trees were also constructed. Also during this step, when I had the sequences, I did domain analysis on them to show the conserved domains and the mutations in the genome. Finally, SVNV proteins were localized basically by cloning all the proteins in the except the L, because L is large, it's almost 9 kb and we couldn't localize it. Also, it's not localized in other studies because it's large and it's almost induced toxicity in the cell. However, we localize N, NSS, NSM, and GN and GC. First, we cloned them in the p-enter d-topo vector, and then they were cloned in the expression vector, which is called p-site, and then they were expressed in the plant cells. In the next few slides, I will explain some of these methods in details. So for the tools that I use for the domain analysis of SVNV, basically I use the TMHMM 2.0 and also the IntraPro, and that's to show the domains in SVNV. After that, I also use the Analyst Mapper to show the nuclear localization signals, which are important for the localization. Finally, the data were compiled using Microsoft Excel and also PowerPoint. So here's was the results on the mutations. So basically, we found that our 33 samples were all positive for the virus, and that's not surprising me because all the samples were collected based on the symptoms of the virus, which were necrosis, and those symptoms were also used for samples for RNA extraction. So I was expecting that most of the samples, not maybe all of them, will be positive. But however, we had 33 samples positive for the virus, and we have a sequence at least 30 samples for each gene. So we have about 30 N, 30 NSS, and 30 NSM. So here is a comparison between each protein with the Tennessee strain. So basically, in the N protein, we found that we have 8 conserved amino acid mutations. So these in black are the mutations that appears in the N. And then here is the percentage of each mutation in the population of 30 samples. These blocks here in red in the Tennessee strain is basically referring to the domains that are predicted or basically that are reported in the N protein. And here I'm trying to show if any of our mutations appears in those domains or not. However, when we look here, those all domains are RNA binding domains, and as I mentioned before, the nucleocapsid protein is responsible for wrapping around the RNA of the virus. Therefore, it needs to have RNA binding domains to be able to identify its RNA in the plant cell. So those domains are very crucial for the virus function or for the protein function. However, when we look here, we don't see any of our mutations fallen in any of these domains, which indicate 2 things here, that these domains are very important and crucial for the virus and maybe cannot be changed. Second thing is that those mutations that we found, some of them were 100%, making sense that they are taking over the population, indicating that they also have other functions that could be self-interaction because this protein have to self-interact with itself and also has to interact with other proteins from the virus and with other proteins from the plants. So maybe those mutations here, however, they don't fall in those domains, which are important, they might also have other effects on protein interactions. When we look in the NSS protein here, we have 6 mutations, and also we have 2 domains in the Tennessee strain. Also, again, when we look at our mutations, we don't see any of them falling in these 2 domains. And these 2 domains are very important here, and they comprise the Walker A and B motifs. And they are basically responsible for the ATP hydrolysis. And NSS, as I mentioned before, it's a silencing protein, and it's very important for the virus. However, our mutations do not fall in any of these domains. But again, as I mentioned before, these mutations were 100%, most of them, indicating that they're also important for the virus, and they might have reflections on other interactions. Finally, in the NSM protein, we have 3 mutations here that are none of them fallen in also in these motifs. So these two motifs are very important for the function of this protein as a movement protein, and both of them are present in our NSM. However, the mutations do not fall in any of these domains. So the phylogenetic trees here, the first one basically is for the nucleocapsid, the second one is for the NSS or the silencing protein, the third one is for NSM. So here's this roadmap. Basically, every sample was red is coming from the north, every sample was green is coming from central Alabama. And when we look here, we can see some similar patterns here. So first of all, the Tennessee strain, which is the first sequence that was sequenced from this virus, when we look here, it's found closer to the north strains, right? And this is for N. But when we look at the also B or the NSS protein, we also find the same pattern. We see that the Tennessee strain is also closer to the north, and it's also the same pattern in NSM. So this makes sense, right? Because when we look at Alabama here, on top of Alabama, we have Tennessee. So because Tennessee is closer to the north, the northern strains are closer to that sequence from Tennessee, which makes sense. So also we use 3 outgroup and orthotypic viruses, including B necrotic mosaic virus and capsicum chlorosis virus, and also the Tennessee strain. And as predicted before, and as we saw in the introduction, SVNV has more closer relationship to B necrotic mosaic virus. We also can see the same pattern in all of our 3 trees. So we see B necrotic mosaic virus is closer to our strains for SVNV from Alabama in N and NSS and also in NSM. Finally, when we look at the evolution of each of these proteins, which also indicated in the mutation that we found, N had 8 mutations as we just saw in the previous slide. And when we look in the phylogenetic tree, it is the most evolved protein in the virus because we can see a lot of groups in this protein that we don't see here and here. So for the domain analysis, we have here N, NSS, NSM, GN, and GC, and L. And here are the domains that were found using the tools that I mentioned before. So this is the size of each protein, and here's the single peptide and the transmembrane domain. So I want to mention that GN and GC, both of them contain signal peptide and also transmembrane domain, and those are very important for the virus and for these proteins to function, and I will explain that in the next slides. I also want to mention that the NLS domain or the nuclear localization signals are very important for the nuclear localization, and also were found in all SVNV proteins. And this is the score of each nuclear domain. And here is the type of each of them. Most of them were bipartite, except L had one that was monopartite. And these are the N-glycosylation sites, which are also important for the proteins interactions. So here, I want to also mention that the value of or the score of the NLS domains are very important to be interpreted in a correct way. So basically, if we have a score between 8 to 10, this means that it's a strong NLS and it will be localized only in the nucleus of the plant cells. Also, when we look here in this value, which is 7 to 7.9, this means that it's mainly going to be nucleus, but it could be other places as well. However, I highlighted this one because all of our nuclear domains, as you can see here, they fall, except this one, they fall in this range. So if it's 3 to 6.9, it means that you will have 2 localization patterns. You will have nucleus and you will have cytoplasm. Finally, if it's lower than 3 score, it will be weak and it will be only in the cytoplasm. So the methods that I use for the localization, as I mentioned before, but here was more details, is that cloning the proteins in the first the p-enter D-topo vector, and then sequence confirmation to make sure that the sequence is good and it didn't change, and also to confirm that the CACC is present in each protein and no stop codons. So these are important checks that we have to do them before we do the p-site. So p-site is a plant expression vector. It basically stands for stable integrative transient expression. So it's a really good expression vector in plants. And here is the site where each of the SVNV proteins will be inserted. So these are the names of the vectors that I use: p-site 2CA and 4NA, 4CA, and also 2NA. What are these names? Basically, each construct will be fused into both C and N terminal of GFP. And then also, each of the proteins will be fused into the C and N terminal of RFP. So basically, this is repeating the same construct 4 times. And why we do this is because to confirm the results. Also because sometimes when you fuse the protein to the C terminal, like here, if you have important domains at the end, it might block it. So this will give you a misleading localization. So when we do all of them, we can then determine which one of them is the most correct. And of course, GFP and RFB are the fluorescent proteins that are going to help us to visualize our proteins under the microscope at the end. And GFB stands for green fluorescent protein, RFB stands for red fluorescent protein. After all these constructs were made, they were sequenced again to make sure that the sequence was good and that they were all placed in the right orientation. And for that, I used the PCR to confirm the orientation. Finally, all these constructs were transformed in Agrobacterium tumefactions. Basically, Agrobacterium tumefactions is a bacteria that can infect the plants and can cause tumors on them. And scientists looked at it and they decided that we can use it to express proteins. Basically, this bacteria here, as you can see here in this infiltration solution, if you look into just one cell and then the plasmid that we inserted through the transformation and it carries the construct or the protein of interest, the bacteria can inject this plasmid into the cell nucleus. And this would allow us to have mRNA of our clone that will be sent to the cytoplasm and be transcribed and translated as a protein. Once it's made as a protein, after 2 days, it basically takes 2 days for this process to happen, we can then look under the fluorescent microscope and see our proteins being localized. I used for localization Nicotiana benthomiana, as you can see here, which is bent. It's a tobacco species, and it's a host for SVNV. So it can be naturally infected by SVNV in nature. So it's a good plant expression for our experiment. So these are the final constructs that were made. For N, NSS, NSM, they were all fused to the both C and N terminal of GFP and RFP. However, for GN and GC, we only fused them to the C terminal of each protein. Why we did this is because we have a signal peptide here, as you can see, and we were careful not to fuse the proteins to the signal peptide so we don't block it, because it's crucial for the function of these proteins. We also, if you notice here, we made two versions of GN and GC, and we call them soluble. So GNS and GCS. Why do we call them soluble? Because if you look at them, you will find that they are missing the transmembrane domain, which is here. So the transmembrane domain is missing from each one of them. Why we did that is because the transmembrane domain, as it's called, once this protein is made, if the transmembrane domain is present in a protein, it will basically trigger the protein and embed it into the membrane. And this sometimes makes it really hard to visualize the protein under the microscope. So we wanted to make two soluble versions to be able to see them. So here's the localization results. Basically, the first localization we did for N. And if you look here, the first column is basically referred to the protein of interest fused to GFP. And we used a plant marker that has nucleus as red. It's called RPH2B. So this is a plant marker, and it looks like that naturally because it's a transgenic Nicotiana benthomiana plant. The third column is basically an overlay between the first two columns. So worded and localized. Interestingly, N localized into the cell periphery and also into the cell nucleus. But that was in the C terminal of GFP. So the N terminal of GFP showed different localization. So this is the C terminal of GFP. Basically the N terminal of GFP, because the protein is fused here to the N terminal of that GFP. And we look here in the picture, we can see that it's similar somehow and also different at the same time. It can also express in the cell periphery, however, it's not localized in the plant nucleus. But when we look in this map that I made here, and I mentioned before that we have nucleolocalization signals in N, and one of them, which has the highest score in these three domains, we found that the GFP might be able to actually block this domain once the protein is being made, and the folding of GFP, maybe it's covering and blocking this NLS domain at the end or the C terminal of the protein. So this localization might not be accurate, and this one is the most accurate because this GFP, when it's fused to this terminal here, it's not blocking anything. So we think that this localization is the most accurate for N. I would like also to mention that the nucleolocalization for N is very interesting, and we were not expecting it, and I will discuss it at the end. For NSS, it localized into the cell periphery and weakly in also the nucleus of the cell. And also N, I would like to say it also had nucleolocalization signals, so that was expected. For GN and GC, because they have transmembrane domains, both of them localize into the membrane of the cell and also around the membrane of the plant nucleus. So there was no changes between these two proteins. Both of them behave the same. Here's our control, which was a free GFP, and as predicted and as it's cited before and referenced, it localized to the cell periphery and inside the cell nucleus. So if you notice, these two figures are missing another protein, which is the NSM, right? We talked about N, NSS, NSM, GN, and GC, but here I only presented N, NSS, GN, and GC. So where is NSM? Actually, NSM was very interesting. So NSM protein, when it was localized, I basically attempt to localize this protein more than 10 different replicates. And every single time, I don't see any protein expression until even 10 days. And that was very frustrating for me because all of them express except this protein. So what was going on in this protein? So later in the infiltration, after 10 days, I started observing patches of dead cells on the plants. And I thought it's dead cells. I talked to my PI and we said maybe NSM is causing toxicity. But we wanted to confirm it with something that show us that the cells are dying with NSM. So we bought this amazing stain that's called Cytox Blue. And basically this stain come in 3 different colors. It can come in blue and red and green, but here we use the blue Cytox stain. So what is the point behind using this stain? Basically this stain is being infiltrated 30 minutes before imaging, and it allows you to see if the cells are dying or not dying. How does it do that? Is basically, if the cells are dying, the cell wall of these cells will not be in a good shape. Therefore, the stain can penetrate the cell walls and go inside and bind to the DNA and RNA. So basically, if you can see this stain inside the cell, this means that the cells are dead or basically having a cell death occurring. My control here was N, which doesn't kill the cells as I localized it before and also can express. So as you can see here, there is no expression. I can say there is no expression because you don't see any green or no GFP. So when I stained the cells with Cytox at 4 days, I started seeing a cell death, and that was confirmed by the stain inside the cells. So in this enlarged section, we can see a few cells here that shows that the stain is now found inside the cells, indicating cell death. But when we look into the NSM of Tennessee, this was the one that was localized from Tennessee. It doesn't have any mutations in it, right? But the one that we have from Alabama, it has 3 mutations. But one of them was this one that we localized it, and we wanted to look into it first before we look into the 3 mutations. So this mutation here appears and happen in this region of the protein. And we wanted to see if this mutation will impact the localization and the expression of the protein or not. So basically, we wanted to see what is the impact of these mutations. And we have 4 steps here to confirm and to study this mutation. First of all, it's to compare the domains of both NSM, the Tennessee, and the Alabama one, to see if any of them will have changes in the protein domains and the folding or not. The second one is to mechanically transmit SVNV of Alabama and see if it can move or not. Why this is important? Because basically, NSM was reported not to be able to systemically move in the plants, which indicate that there's some problems with the NSM of this protein, of this virus. But also that was conducted about 15 years ago, but we don't know if it can move now or not. So conducting this experiment would show us if SVNV can move or not. And this will give us ideas if NSM can work or not. The third thing is to localize NSM of Alabama, which is this one. And then finally is to compare between Tennessee NSM and Alabama NSM using the Cytox to see which one of them is causing early cell deaths. So to check the first one, I used the motif finder and also the entry protein to look into the domains of both of them. So we have the tubule formation domain, which is called 3A, and it's predicted because NSM move the virus from cell to another cell using the tubule formation, which is a fascinating mechanism. And also it contains a very interesting domain that is called DUF, and it stands for Domain of Unknown Function. This domain has never been found in any virus before, as I checked it so far. It only has been found in bacteria, specifically Clostridium species, which are known to produce toxins. So that was very shocking to me. We don't know the function of this protein yet. It doesn't have a assigned function. So we have a mutation here, as I mentioned before. So what is the impact of this mutation? So I looked into the folding of this protein using the AlphaFold 2 tool, and it shows something very interesting. I was not expecting to see any changes in the folding because it's just one mutation, right? But that was actually not true. Because when you look into Tennessee NSM, it's not folded to itself, it's open. And that's a problem with the protein. Usually proteins like that, when they're not folded to themselves properly, they will trigger bad things in the cell and they will trigger cell death and they won't be functioning as well. And that maybe indicate why we couldn't see NSM of Tennessee in the plant cell. However, NSM of Alabama, with one mutations, it was able to fold to itself and have a proper folding. And that may indicate that this protein might actually express in the plant cells. So the first check was done. Second is to mechanically transmit SVNV. So basically, I used these methods in the picture here. I used infected plants from the field, which were soybean. I ground them in this extraction using a specific buffer with a specific pH to help the virus not to be degraded, and then inoculated the plant on bins and then kept it for 14 days and watered it normally. After that, I started observing symptoms, as you can see here, on the top of the leaves. However, the inoculation was done on the bottom or the lower leaves of the plant. So this indicates that if you can see symptoms on top, the virus has moved up there, so the virus can move. So good so far. So when I use the PCR to determine or to detect if the virus can be detected on top or not, I found that using the N protein, I was able to detect the virus on the top leaves, indicating that the virus now can move systemically in bend. And this is my positive control and negative control. So the second check was done. Good. The third one is to actually localize NSM. And I was really happy because I finally can see NSM expressed in the cell. Before that, as I mentioned, I did 10 replicates for the Tennessee NSM, and it never expressed. Now this one with one mutation can express, which is very interesting. It both localized into the cell periphery and into the nucleus of the cell, which also agrees with the NLS predicted domains. So third one was done and the protein can be expressed. Cool. Let's move to the final comparison between NSM of Tennessee and NSM of Alabama. Okay, so here I put them both together and I took images at 2 days post-infiltration and images at 4 days post-infiltration. So the first one, as you can see here, it's not expressing the NSM of Tennessee. NSM of Alabama is expressing normally, it's fine. My control is also expressing. So I also use Cytox. I did the same. At 2 days post-infiltration, we can see that the blue stain is still outside the cell wall in all of them. So basically, there is no cell death at 2 days. But when we go to the other side at 4 days, we can see here in the NSM of Tennessee that we have blue stain inside the cells, as I mentioned, indicating cell death at 4 days. However, NSM of Alabama does not cause cell death until 4 days. Would it still cause cell death? I would say yes, but maybe after late in the infection. And this is very helpful for the virus, because if the virus can kill the cells early in the infection, that's a problem. Because the virus now then cannot move systemically because it killed the cells. It might not be able to leave the cells. But if it kills the cells late in the infection, where the virus have already moved from these dead cells, then this would result in more successful infection for the virus. So cool so far. Okay, so let's look into the localization of the proteins in insect cells. So also this picture was done by my previous colleague, Michael Mayfield, and basically he localized all the proteins in the insect cells. And we want to compare them because, as I mentioned, these viruses are cool because they can both infect the plants and the animal system. And the animal in this case are the insect, which are thrips. So we use SF9 cells, and the N protein localized into the cytoplasm, forming a big cluster in the cytoplasm. However, this agrees actually with the localization in the plant in the N terminal, but we did not attempt to localize in the C terminal, because in the plant it localized in the nucleus, if you remember. So we didn't attempt to localize it in the C terminal of GFP, but I would expect that if we attempt that, it might be similar to insects. However, when we look into NSS, which also was localized in the same terminal as the plants, it localized both in the cytoplasm and weakly in the nucleus. You can basically weakly see it in the nucleus here, and it more appears when you overlay. So what is this blue here? Sorry that I didn't mention before. It's the DAPI stain. So the DAPI stain basically stained the nucleus. So we can confirm if we have something in the nucleus or not. When you overlay them, you see a change in the color here. It's a slight change, but indicate that NSS is present in the nucleus, but weakly in the nucleus. It might not be retained complete in the nucleus. It might just be there for a few days until it does a specific function that we don't know yet, and then it leaves the nucleus. NSM, and this is the NSM of Tennessee, not the NSM of Alabama, it was able to express an insect, but it didn't express in the plants, okay? So NSM of Tennessee was able to express normally in insects and form these bright aggregates in the cytoplasm. And that actually gave us some ideas about how this protein might be functioning, and if NSM can function in insect, the original one, but not in the plants, maybe this virus was mainly coming from insect and it just happened to infect the plants and adapted to it more and more and more until it was able to evolve an NSM of the current isolate can express in both plants, and I expect it will also express in insect one if we do it. When we look into the GN and GC, we see that GC is absent from our figure, and that's because GC was really hard to localize in insect cells, and my colleague Michael did not or couldn't take pictures of that because it never expressed. So basically we end up having just GN, and we have two forms of GN the full length and GN soluble. And this is what I mentioned before. GN has a transmembrane domain, so it localizes in the cytoplasm in the membrane of the nucleus as well. You see this thin line around the nucleus, this is the nuclear membrane. However, GNS, now it's soluble, doesn't have transmembrane domain. Now it can localize in the cytoplasm and be spread more and more soluble. Again, our free control is here, which is a free GFP in insect cells. Okay, we also conducted co-localization. So what is co-localization and how is it different from just localization? Co-localization, as you can tell, we are localizing two proteins from the virus in the same time. So this basically to show us if we have any changes in the localization or not, or basically if any of the proteins, if the presence of it will affect or change the other one or not. And the main idea here is that when you overlay green with red, you get yellow, right? So here, the column in the middle, when we have overlay, whenever you see yellow, it means that they basically co-localize in the same spot. Why this is important? This gives us ideas. It does not confirm it, but it gives us some ideas. If the protein, both of them, are present in the same location, that could indicate that they might interact. We cannot, of course, say they interact here because we have to conduct a different mechanism or different protocol, which is using pull down or biopsy to confirm that they really interact or not. Now, protein interaction is very important to fully understand how the virus is functioning, and this is the next steps for us. It's not included today, it's the next. Also, when I did the co-localization, let me take you back here. So all these images were taken at 2 days post-infiltration, okay? All of them didn't look different. They didn't look different after 2 days, except one combination, the combination of NSS and N together. After 2 days, they started showing different localization as seen in 2 days. So let's go back to this figure here. Basically, I wanted to document this change, so I basically localize NSS alone, and then N alone, and then I put them together. When you localize NSS alone, as mentioned before, it localizes to the cell periphery and weakly in the plant cell nucleus, as you can see here. However, at 5 days, it starts to have more aggregates outside the nucleus, as it was also hypothesized before. N protein localized in the cell periphery and inside the nucleus until 6 days. It was always soluble to the cell periphery and into the cell nucleus as well. However, when we put them together, we see changes here. So at 3 days post-infiltration, actually, let's talk about 2 days. At 2 days post-infiltration, we can start seeing voids around the nucleus. So both proteins are present inside the nucleus, NSS and N, as you can see here. However, we also can see voids, or basically empty bottles outside the nucleus. And that was very interesting. And we don't know what is the function of these voids and why they are being formed. However, at 3 days post-infiltration, we can start seeing NSS being more soluble and also aggregates in the cytoplasm as small clusters of aggregations. Now, by aggregation, I mean that the protein units will bind to itself and form chunks of proteins that it moves around the cell. And this is what we can see here. It was actually very beautiful to look at it. It looks like almost a Christmas tree that is really bright, but we can start seeing that NSS is behaving differently here. N was still soluble. However, at 4 days post-infiltration, N have been shifted to complete aggregation outside the nucleus, and this was consistent until 6 days. Also, NSS followed the same pattern and started to form more aggregates outside the nucleus. So when we look basically at 6 days post-infiltration, both proteins are present mostly outside the nucleus and very, very close to the nucleus. And this might mean that this virus needs to replicate very close to the nucleus. Because these two proteins present outside the nucleus, it might indicate that they replicate there. And of course, this is all predictions, and we have to show more tools and more results to confirm that. So what is the summary of this work? Basically, the data that we got from the sequence and from the localization support our hypothesis. First, our first hypothesis was that SVNV genome is evolving since it was discovered 15 years ago. And we have seen about 18 mutations in N and NSS and NSM protein, indicated that our hypothesis was right. So now someone would ask me, what could be the advantage of these mutations on SVNV? Well, it can help the virus fit more in its environment. Usually when viruses or microorganisms or everything, when they evolve, they mostly are trying to have a better fitness in their environment, to fit better and to be able to overcome diseases and be better in the future. And that could be one of the reasons SVNV is mutating. Also, as I mentioned in the background, SVNV has a very narrow host range, which means it's only infecting few host range. So maybe this helps the virus to move from plant to another plant. And that was seen in Alabama this year, because we detected the virus for the first time in peanut, and it has never been found before. So this means that the virus now can expand its genome by mutating, because when the virus mutates, it can adapt to different hosts. So the second hypothesis that we also hypothesized was that the localization of SVNV might be similar but not identical to other orthotype viruses. And here I picked the most two divergent orthotype viruses that were localized. So we have CACV and TSWV. So CACV, for its N, it localized outside the cell nucleus, but very close to it, and also to the cell periphery. TSWV localized in the cell periphery, as you can see here, with some aggregates. Unique to SVNV, we have nuclear localization in the nucleus and also cell periphery, which agrees with both these two viruses. However, the nuclear localization was unique to SVNV and protein. For NSS, it was seen in the nucleus only in CACV, and our SVNV agrees with that. It has a similar localization. But for TSWV, again, it localized into the cell periphery and also around the cell nucleus, as you can see here. The NSM protein, as a movement protein, it basically localized in the plasmodesmata, which are small tunnels that connect the plant cells together. And as I mentioned, the proteins of orthotypoviruses, they have the tubule formation domain, which function as mitigating the PD size to allow the virus to move from cell to another cell. So it was always being seen in the plasmodesmata and CACV and TSWV. However, our NSM of Tennessee did not express and kill the cell of the plants. However, it was expressed in the plant in insects. But when we localized the mutant of Alabama that I discussed before, it was able to localize into the cell periphery, maybe the plasmodesmata. I haven't tested that yet. I will have to use a different stain called aniline blue to show if the proteins are found in the plasmodesmata or not. And also, it was found in the nucleus as well. So far we have 3 proteins in the nucleus, N, NSS, and SM. And this agrees with their nuclear localization signals. For GN and GC, they matches TSWV, where it localized into the cell membrane and also the nucleus membrane, as we can see here. However, for CACV, it localized into the cell membrane and also inside the nucleus. I would like to mention that in CACV virus, they localize both of them in the C terminal of the GFP, which we didn't do because, as I mentioned, we were not trying to block the signal peptide. So these localization results support our hypothesis that SVNV have similar and unique localization. So now we can understand somehow why SVNV is clustering alone with binocrotic mosaic virus, because the sequences that we presented today in this talk have variations and they have mutations, and that led to localization pattern to be also changes that have some similar localization and unique localization to SVNV. And now we can understand why SVNV is a cluster in specific group here with binocrotic mosaic virus. In the future, it will be very interesting to reveal the relationship between these two viruses, because I believe that they have some kind of association, and to understand how binocrotic mosaic virus behave similar or different to SVNV or not. And finally, this work is published. You can now review it, and I would like to have any questions from you. And future the directions is that basically for the NSM protein, I localized the Tennessee, which was the original one, the wild type, and also I localized this one that was just the last mutation here. And both of them, this one expressed, but this one does not express. But now we have 3 other versions of these mutants. So we have the first mutant that carries just the first mutation. We have the second mutant that only contains the second mutation, and we have the last mutant that carries 3 mutations together. And it will be of interest to localize all of them to see which one of them exactly will be expressed or not, because For example, if this one can express that I showed in the talk today, but these two cannot express, this means that this one here fixed the domain, or like modulated the domain that was responsible for the necrosis. So this will be interesting in the future to know what's going on. And actually, I've been working on this and I got it done so far. But I can tell you right now is that both of these mutations, they can also express. So finally, and after 50 minutes of talking, I'm sorry for talking too long, but for acknowledgement, I would like to especially thank Dr. Sakura for collecting all the samples for us, and a special thanks to our amazing PI, Dr. Martin, for supporting this work by advising and commenting and fixing all the errors, and also for the funding agencies that are placed here in the bottom: Auburn University, the Alabama Soybean Producer Association, and also the Alabama Peanut Producer Association. I would like to thank Martin Lab members, including all these amazing people working their master's and PhD, and some of them are undergrad. And thank you so much for your listening, and I'm ready for your questions.
Thank you for following us. We sincerely thank you for your continued support and engagement across all our platforms. Your trust means everything. Stay connected. Together, we grow.